Welcome everyone to part 2 of our biomolecule lecture. In part 1 we covered carbohydrates and we covered lipids, and in part 2 we're going to cover proteins and nucleic acids. And we're still answering the question of what are the little parts that build up both our cells and us. Let's get going. We're going to start here with mentioning our first biomolecule, which is proteins. And proteins have a hugely important impact on our lives. They account for more than 50% of the dry mass that most cells consist of. And what we want to do is we want to take a look at their functions first before we get to their actual structures. So here is a list of all the different proteins that we find and what they do. And it goes from enzymatic to structural, storage, transport, hormonal, receptor, contractile, motor proteins, and defensive. Whew, that's quite a list. All right, so let's go through these and let's talk more specifically about what these jobs actually are. So in our last lecture, we mentioned enzymes, and we said they were little protein helpers that allowed chemical reactions inside of organisms to take place at a quicker pace, okay? So we can see selective acceleration of chemical reactions. All right, here's an example. In your body, you have digestive enzymes. We're all well aware of that, okay? We know that when we eat food, it goes in one way, and through various processes, mechanical and chemical throughout our body, the waste leaves our body in a different way, and our body uses that food in a different way. It doesn't stay the original molecule that we were eating. So enzymes have the ability to take apart certain types of the food that we eat and turn it into something that our bodies can actually use. But without those enzymes being present, being able to do that job, we don't obtain optimal nutrition enough to even survive. It's because of these enzymes that we can break down food and actually use them by the cells of our body. We have structural support proteins. These literally will support the cells and some larger structures in our body from falling apart. Okay, so if we looked at a structural protein like collagen and elastin in animal connective tissues, basically we're talking about here what you find in cartilage and tendons and ligaments, and in skin that helps with elasticity, keratin in hair, horns, feathers, and other skin appendages. And we even have some of these that run through our bones. So collagen will actually work as kind of like rebar within our bones themselves, giving them additional strength and making them incredible structures to hang, muscles and organs off of. We have storage proteins. And here you can see a few examples like egg whites. Perhaps people eat eggs for the amount of protein that they have. We have the protein of milk. We have storage proteins in plants and seeds. And it all comes from the storage of what we call amino acids. And as we get into structure, you're going to see the connection between amino acids and proteins. We have transport proteins, which help move stuff around within organisms. Use an example, hemoglobin. So hemoglobin is a protein inside of our red blood cells that moves oxygen and carbon dioxide from where they're picked up to where they need to be released at. We have hormonal proteins, which coordinate an organism's activities. And the one that's highlighted here is insulin. So insulin is a hormone that your pancreas secretes after you eat a meal of food. So ultimately that meal will contain the nutrients that will eventually get us the energy that we need. So the insulin will provide a hormone that reaches all cells, and it tells those cells to open up and allow sugar to rush in to your cells so it can be properly nourished. And without insulin, a protein hormone, we can't do that, and our blood sugar levels would be completely out of balance. We also have receptor proteins, and these are going to respond cells to certain chemical stimuli. And we can see the example is receptors in nerve cell membrane. So imagine that you have a cell, and on the outside of this cell, you have these little proteins that are embedded, and they're kind of like gatekeepers. So they allow some things in and they keep other stuff out based on your cell's needs. So these little dots here would be those receptor proteins. Contractile and motor proteins. These function in movement. So if you flex a muscle right now, what you have are two proteins inside called actin and myosin, which are actually causing the contraction of that muscle fiber. Some of our cells can actually move around, and that's due to proteins in the little hairs that cover their surface. And I'll draw a few on this cell here. So little hair-like structures that beat in coordination can propel a cell in a forward direction. And some cells actually have a whip-like tail that actually moves in response to the energy that it possesses towards stimuli or away from stimuli. And ultimately the flagellum itself is a collection of proteins that work to propel a cell forward. And then defensive proteins, so important right now at this point in 2020. These are going to help protect against disease. So we have these special workers in our body called antibodies that combat bacteria and viruses. Okay, now these antibodies serve a special function to recognizing bacteria and viruses, and then ultimately binding with them to take them out of commission, to basically target them for destruction. Okay, when an antibody links on to a bacterium or a virus, it then is a signal to your immune system, hey, we got to get rid of this. So it's so important as a defensive measure to have these protein antibodies in play. So what we can say with this gigantic list of functions that are functionally and structurally diverse. They do a lot of different things. That's why they're so important. Here is a picture of proteins, and this picture, you can see there's a lot going on here, is actually going to be the 4 levels, which we'll talk about a little bit later with these proteins. And it starts from a primary structure, and we could call that level 1. Okay, so let's go ahead and I'll get my pen going here. We'll call these guys here, and I'm going to take that back for a second. So these are level 1 proteins. What I mean by that is that on the simplest level of protein structure, you see these 3-letter combinations separated by these dashes. These all represent what are known as amino acids. Amino acids are the monomers that wind up as your simplest piece of your protein. And we talked about monomers at the last lecture. We said it was analogous to a train. The train was a polymer, and each train car was a monomer. Well, here in the primary structure, each amino acid is actually the monomer for something bigger, which will be a polypeptide when we get to that level. Now, our level 2 on our way to a workable protein is going to be these two. We have what's known as an alpha helix and a beta pleated sheet, or a beta sheet. And you can see right here that this kind of has one of those coily Q types of shapes here, and that is what these amino acids do when you get enough of them in a line. They start folding themselves based on hydrogen bonding into these shapes. Now this one here is much more gradual. This is a sheet, so it's flat. You can see hydrogen bonds that are holding it together, and it's a much more wide platform for these amino acids than what we find in the alpha helix. When we go to level 3, which is known as the tertiary level, we can see that these coils are now something smaller in a bigger picture. So here we have 2 of those coils, and this region here would be more of those beta pleated sheets. So we bring it all together in one big polypeptide that has multiple parts in level 3. And then to make a workable protein, we have to reach level 4. Okay, so level 4 is what we call the quaternary structure. And here you can see this structure here, but now we also see this one adding on to it. And you see right down the middle here, this is where those 2 pieces are going to bind together to make the largest structure of protein we can have at level 4. So this right here is a workable protein, something that has a job within an organism's body and does it. This picture here, let me go ahead and erase. Let's see if I can do that real quick. Here we go. I'm gonna erase the pen. But some of the standard code for proteins in biology books is big purple blobs, okay? And I mean that lovingly towards our proteins because they're so important, but they look like big purple blobs. And each one, you'll notice you have 5 of them right here, kind of embedded in this thing. This is a cell membrane right here, and you see the receptor proteins and these channel proteins, these gated channel proteins, they go span directly across. Many of them do that. Some of them don't. Some sit on the outside, some sit on the inside, but these are, we call them transmembrane proteins. They go all the way across. And you might be able to, by the look of them, predict what their jobs are, okay? So when I see something spanning a membrane that has a channel going down it, I tend to think, wow, this is probably for flow of things from the outside to the inside to the outside. This one, same thing, but there seems to be a flap on this one, so maybe it has a little control over it. This one here is squeezed shut at the top, open at the bottom. So logic tells me these are likely going to allow things to pass through, okay? Now over here you see a receptor protein, and this one over here, it is one of those that catches things. And this one would catch a little molecule right in this little nook right here. Maybe it's a triangle shaped molecule. And by catching it here, there's a chain reaction within the protein and it signals something down on this end to happen, okay? That's often how receptor proteins operate. Over here we have a protein that's snapped shut at the moment. And you can see above it we have carbohydrates. We remember these from our first lecture. They form branched chains like this. And this one serves as kind of like a recognition protein. So it holds out this little sign, and this sign might be like a chemical sign for you, okay? Y-O-U. Maybe all of your cells have this special carbohydrate chain that tells your body and your immune system, hey, don't attack me, I'm part of you. And it's like a recognition site. And then perhaps there's some trigger that makes this thing open up, and then a flood of ions might flow through here. So I don't want to forget about these purple guys down here. It kind of look like spiders. These are some of those structural proteins that make up what's known as the cytoskeleton. So even cells have a cytoskeleton. Now it's not made of bones, but it's made of flexible protein material that not only keep the cell in terms of in a non-collapsed form, it's spread out, there's volume to the interior, but it also anchors some of the parts of the cell inside here in this beige area in place, and doesn't allow them to squish over to another part of the cell where it shouldn't be. All right, let's go through some protein basics and terminology now that we've seen a superficial picture of them. They consist of one or more polypeptides, and polypeptides are going to be polymers, like we've mentioned in our last lecture, built from the same set of 20 amino acid monomers. So remember, a monomer is that building block, that unit, and there's 20 of them that make up every protein, okay? Now, the amino acid itself, and that's what this is down here, it gets its name from a couple of functional groups that we find on it. So every amino acid on one side will have what's known as an amino group, and on this side over here, a carboxyl, or sometimes called a carboxylic acid group. Every single amino acid has this and this identical, and even this little H on top. Now, the thing that's going to differ amongst each of the 20 amino acids is this thing here called the R group. So the R group, you could think of the R as meaning like rest of the molecule. The rest of the molecule may differ from one to another in your 20 amino acids, but this, and this are identical in all 20. Okay, so amino acids are organic molecules with carboxyl and functional groups. We just saw those. They differ in their properties due to side chains called R groups. And again, you can think of R groups as rest of the molecule. Okay? All right. And if we go ahead and we take a look at this picture, it suddenly looks a lot more complicated, right? But let's take a look at what we have here. This purple box was basically what we just explained on the last slide, was the same for every single amino acid. So this is amino acid 1, this is amino acid 2, this is amino acid 3. Now, they have to be flipped upside down. So the R group is this, okay? The rest of this, notice, is identical, right? Here's another R group. That one's unique looking. this hexagon looking thing here. This one's unique looking. It's different from those two. So these are 3 separate amino acids. And to go from monomer to polymer, what we have to do is we have to form something called a peptide bond, okay? Now, what's good for us is that mechanism for forming a peptide bond is shown right here. This should look really familiar to you right now, okay? Hanging off of one end of a molecule is an OH. Hanging off the other end is an H. And they find each other, they collide with each other. And what happens? Water comes out. So what did we call this when that happened? What type of action connects 2 amino acids together in a peptide bond? You just saw it with both carbohydrates linking together, those monosaccharide linkages, and then in lipids, we talked about glycerol binding onto the fatty acids. And if you're thinking condensation reaction, you got it. A condensation reaction, we call it dehydration reaction, is when a water's removed and this leftover bond here and this leftover bond here connect together right here. Here it is, your peptide bond. Okay, and that's how you go from, this would technically be called a dipeptide because there's 2 amino acids. We add a third one, we have 1, 2, 3, we have a tripeptide now. Okay, so a polypeptide is a polymer of amino acids. Polypeptides can range in length from just a few to more than 1,000 monomers. Imagine a chain of 1,000 of these amino acids in a row. It's pretty awesome. The other thing I want to mention at the bottom here is that each polypeptide has a unique linear sequence of amino acids. Okay, so we can see that this one here starts with your number 1, 2, and 3. Do all proteins start with these 3? Nope. Each one is different in its own unique way. You can put any of them in a line together through peptide bonding, and sometimes you could have an alternate pattern of one then another. Sometimes you could have, well, I can imagine you could have all 20 in a row. You could have some repeat themselves. There's a lot of variety in your polypeptide sequences as well. So how do we get function from its structure? Well, each protein is going to ultimately have its own shape. You can see here there's some curly Q type of shapes in here. You have some ribbon flat areas out here. And that unique shape is there for a reason. There is a purpose for it that has evolved over time. Let's look at this one, for example. This special protein here, this is the ribbon model of it. This is a scaled down version of what it looks like. If you use a space filling model that uses spheres for atoms, you can see it's kind of like a big blob, as we said before. And this has a function. Its function, when it's folded in this way, is a bacteria buster. It finds certain bacteria and breaks up their cell walls. So this one here helps you, lysozyme. And you're going to find it present in many secretions from bodies. Tears, mucus, saliva, and breast milk. Basically, this is going to be present where you have a lot of holes on your body. Why are they there? Because the holes on your body are places of bacterial and viral entry. That's where the bad stuff gets in. So having a designated army that works there that kind of takes apart those threats to our body is very important. And this specific shape that it has is designed to bust those bacteria. Let's look at another example. Here we have what's known as an antibody protein, one of those helpers we talked about at the start. And here we have a protein from a flu virus. Now, if you look at the two shapes, something should stick out. Okay, there's a lot of lines and a lot of chaos going on here, but look down the middle here. Notice how this shape on the left seems to fit this shape on the right. It's kind of like that analogy in earth science when you learn that the eastern coast of South America looks very similar to the western coast of Africa because at one time they fit together. So this protein and this protein, one from your body and one from a flu virus, fit together. And that's important. This structure determines its function. By binding these two together down the middle, you now made it a target for your immune system to come in and destroy it. And that's going to keep you healthier if you have a mechanism that takes out things like viruses and bacteria. And this very, very specific antibody's job is to find this flu virus, bind onto it, mark it for destruction, and then the immune system takes care of the rest. In our last lecture, we also talked and brought up enzymes as a topic, okay? Enzymes were a protein, and we said they helped to break down carbohydrates faster, okay? So what we can say is that they act as a catalyst to speed up chemical reactions. All right, let's figure out what's what on this picture before we move forward, okay? Big purple blob codes as a protein. And this one has a job. It is one of those enzymes, and its name is sucrase. Now, just a little FYI, when you see ASE behind some letters in an enzyme's name, it typically means that it's going to work on what comes before it. And here you see SUCR, which is the beginning of the word sucrose. So sucrase, this big purple blobbed enzyme right here, is going to take apart sucrose, okay? So let's see how that happens. Let's imagine you eat a good lunch and there's some sugar in it. So there's some table sugar in it. And here it is. Here's your table sugar, sucrose. And you can see it's going to nestle in these two little active sites, these receptor sites of sucrase. They're going to fit right in there. And then from this picture to that picture, something happens. Can you see the difference between this and this? Okay. I hope you're zeroing in on the fact that the bond between these two is missing. Now, what happened to go from here to here? Well, let's look. Water came in. So if water comes in and disrupts a bond or breaks a bond, can we remember what that process was called? Hopefully you're remembering the reverse process of dehydration synthesis, which was called hydrolysis. Hydrolysis was the breaking of a molecule using water. Hydro is water, lysis breaking. So we're breaking this molecule by letting the water attack this bond right there, and now it's gone. And these two are no longer connected to each other. Well, what are these two? Well, this one's glucose. I'll put a G on it. This one's fructose. And what do? They float away. They leave this enzyme and they float away to be used by a cell somewhere. And meanwhile, the enzyme then is emptied again and it's ready for more sucrose. So enzymes can perform their functions repeatedly. It's not one and done. You can load this up and do it again and again and again until your blood sugar has ultimately gone down because you've broken all those sucrose molecules, turning them into glucose and fructose, and now your cells open their doors, let them in, and your blood sugar goes down. So very, very important function that this enzyme called sucrase has. A little bit more about the protein structure itself. We're revisiting that from one of our first slides. We said there are 4 levels of protein structure. First level was called the primary structure, and that was its unique sequence of amino acids. So amino acids, there's 20 of them, and which one comes first? Which one's second? Which one third? Which one fourth in that long chain? That's your primary structure. The secondary structure were those coils and folds that we saw, okay? And we'll revisit those in a minute as well. The tertiary structure is determined by interactions amongst those R groups. Remember we said they meant rest of molecule because everything else was the same in an amino acid. So when this growing polypeptide starts folding upon itself, tertiary structure will start revealing itself as well. And the last level was quaternary structure, and that results when a protein consists of multiple polypeptide chains that are bonded together. All right, let's dive in. This here is your primary structure. You can see the sequence is numbered. Here's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It just goes on and on all the way to 25. And you can think of them like letters in a long word. One letter has to come first, then another one, then another one. And there is a directionality to it as well. So you can see that these are 3-letter combinations, but what do they actually mean? What are the amino acids? Well, they all have names. Like this one is 3 letters short for glycine. This pro is proline. There's threonine. There's glycine. So each one has its own name, and you'll see some of the same ones again. You'll see some new ones. Here's leucine. There's methionine, valine. Again, there's 20 different names of them. So a question I have is, why do you think that they are in this order? Let's say your body needs a lot of this growing polypeptide right here. Why does your body know how to build this very specific one? What ultimately gives it the direction to do that? And the answer, perhaps maybe you had biology in an earlier grade, maybe a middle school. It goes back to the original instruction list for building any living thing, which is your DNA, okay? Your DNA ultimately has the instructions to build up anything you need when you're out of balance. And if it's this polypeptide you need, the DNA ultimately will be the code that translates into this polypeptide, starting here with amino acid 1 and finishing here with amino acid 25. Pretty amazing. The secondary structure, as we mentioned before, has two possible shapes to it. We have alpha helixes, which you can see down here. You see it's very busy in this place here because these are basically linked amino acids in repeating units, one after another after another. And every time you see an R, you found a new amino acid. So they just kind of very tightly coil amongst themselves. This purple box here is an example of just one. And then there's another one here, and then another one here. And same thing with a beta pleated sheet, which is our next shape. You can see real gradual folds here, not like coils like we see in the alpha helix, but very gentle. Another thing to see is these H to O's, O's to H's. What were these again? Well, these were hydrogen bonds. These hold these coils in place. These hold these sheets in place. So they're very important the way they line up, so the right H's can bond with the right O's and keep that structure sound. So the coils and folds of secondary structure result from hydrogen bonds between repeating, we'll just call them units of the polypeptide backbone. All right. Tertiary structure. This is determined by interactions between R groups rather than interactions between backbone constituents, which are just basically your amino acids. So what we have here is, if you remember those R groups, they are going to be present in a lot of the shapes that keep this, what ultimately is a big old protein blob, in this particular shape. For example, look at this loop out here. This loop is permanently here. Why? Because there are interactions between the R groups that keep it extended out there. This space here probably has a bridge holding it apart from the other side. We don't want these touching each other for this protein to work properly. These coils here are probably held in place by a series of chemical interactions that keep it in that position only. So the interactions on a very chemical level are between those R groups. We know what hydrogen bonds are. They're in there. We know what ionic bonds are. We've learned that. They're in there, positive and negatives. Hydrophobic interactions are basically where you have two nonpolar R groups interacting with each other. In science, we say that like dissolves like. Sometimes we say birds of a feather flock together. So if you're similar chemically, you have no problem hanging out with other things that are chemically similar to you. And then there's another one called van der Waals interactions, where it's just like these brief momentary attractions. And then sometimes you have some real heavy duty bonds on the inside called disulfide bridges, okay? So right here, this is a disulfide bridge. Notice you have two sulfurs. That's where the disulfide comes from. So it's like an anchor. So maybe there's one here and here holding those two apart. And you find those with the amino acid cysteine. When there's one on each side, they form disulfide bridges, keeping an area of that protein extended out. This is a closer look at some of the other interactions as well. So our purple is our polypeptide backbone. So imagine, we'd have amino acid 1 here, 2 here, 3, 4, 5, 6, we'll say 7's down here. And you'll notice that there's all these things going on between the folds, between the curves of your polypeptide backbone, like hydrogen bonding. Here we go. An H and an O, which are attracted to each other, keeping this part connected with a little bit of force. We have our ionic bond here at the bottom of this curve, a positive and a negative. We know they attract. We have our disulfide bridge. So over here, over here and over here, this is locked into positions. This loop stays a loop. And then up here we have hydrophobic interactions. Again, when you have a whole bunch of non-polar stuff like on this amino acid here, and notice that there's another one, the same one down here, and their R groups are interacting together. So they have no problem basically hanging out together in this inner part of that loop. So there's a lot of chemical forces going on inside of these to make them the shape that they are. All right, we are to tertiary structure at this point. Here's a whole bunch of alpha helixes right there. Here's a whole lot of beta pleated sheets, some open areas as well. And now this is a tertiary protein, or tertiary, we call it polypeptide, not an official working protein yet. And it's going to come over here and take this position. But notice you also have this one here, this one in the back, and this could be an extra one there as well. And it's not until they're all together we reach that fourth level, quaternary structure, which ultimately is your working protein. When they all come together in that clump, they then begin to operate. Okay, so quaternary structure results when two or more polypeptide chains form one macromolecule. And there's some examples that we know very well from just human biology. Collagen, for example, that's a fibrous protein consisting of 3 polypeptides coiled together like a rope. So you see a lot of that alpha helix going on within collagen itself. And you can see it's the main component of some of your body tissues. Your tendons hold your muscles to your bones. Your ligaments hold bones to bones. Cartilage at all your joint areas and in your spine. It's part of your skin, helps your skin stay elastic and tone. And then it's found even in bones as a reinforcing protein in the calcium matrix of your bones. And then over on the right, you can see a more we'll call it spherical shaped one here. This one is called your hemoglobin molecule, and it's globular. You can see it's a big round glob or blob. Consists of 4 polypeptides. 2, we call these alpha chains, and 2 beta chains. And this is so important, this protein, because ultimately, as a red blood cell, let's imagine this big circle I drew is a red blood cell. O2 at your lungs has to load up onto these little what we call heme groups, and there we go. And CO2, carbon dioxide, your waste product, has to load on when that oxygen reaches its destination. So the oxygen is picked up at your lungs, and then it's carried all over your body from the top of your head to the tip of your toes. You need oxygen in every single one of those cells, and the hemoglobin is going to do that job for you. So that protein is transporting. And meanwhile, those same cells are probably trying to get rid of their carbon dioxide waste, so they load it onto the hemoglobin as it takes its return trip to your lungs to, again, add oxygen to it. Super important. Now, hemoglobin itself, there's a lot of it per red blood cell. So think for every red blood cell you have, there's hundreds and millions of these hemoglobin molecules to load up a lot of oxygen and be efficient. Our fourth biomolecule is the nucleic acid group of molecules. And our most famous one, I think you can tell from the picture what we're seeing here, is DNA, of course, right? So DNA is our most famous nucleic acid. And all of those amino acid sequences that we saw in the protein section is programmed by your DNA. Okay, and that DNA has little tiny portions called genes. So imagine if we could take a piece of this piece right here, you can see it's been bracketed out for you. This stretch of DNA codes for a protein like we saw in the last biomolecule. These are the instructions here to build one of those. So genes are made of DNA, just one type of nucleic acid. And its primary role, its function, is for storing genetic information. So you are basically about 25,000 genes of information that you received from your mom and dad. Your mom and dad stored that genetic information in their DNA, and they passed it to you. And you have a combination of both your mom's genes and your dad's genes that produce that very unique you. And you, as being super unique, will now pass on half of that content to your offspring if you choose to have offspring one at a moment in your life. Some other roles of nucleic acid. Well, think of every time you have an injury where you get cut or a wound. What you're going to need there is you're going to need cells to fill up that gap, okay? Because you've literally broken the surface of your body and you don't want to have a scab forever. You want to have cells there. So we want to have the right types of cells there as well. If I have an abrasion on my skin, I want skin cells filling up that open, empty area. And DNA has to be copied into the new cells for that to happen. We have to know what type of cell to put there in that hole in your body. So DNA has to provide directions for its own replication. And in doing so, you can then heal up from wounds with the right types of cells. Okay? Now, DNA will direct the synthesis of RNA and through it control protein synthesis. Okay, so DNA is going to be related to this other molecule, this other nucleic acid called RNA, which is like a copy of it. And the RNA copy will ultimately get you a protein polypeptide like we saw in the last one. So it goes from DNA to RNA to protein. Okay, now I want you to think of RNA kind of like a copy of DNA. So imagine I had to copy a textbook page for you guys. I'd take it to the copier, I'd hit 125, and I would make 125 copies of a single page out of a large book, okay? Then I'd give it to you, and you would do something with it. You would do some synthesis with whatever page I gave you, okay? Some learning from it. And it's kind of that way with this DNA, directional flow. From your original blueprint of instructions to copies of those instructions, and then translating those instructions through protein synthesis. That's how it occurs. We call that the central dogma of biology. We also use nucleic acids as energy transfer molecules. So the reason why you're thinking right now and writing right now is because you have a special nucleic acid energy transfer molecule called ATP. Lastly, there are some other nucleic acids inside of organisms that work as intracellular signal molecules. So perhaps something on the inside of the cell needs to communicate with another part in the interior of the cell, and nucleic acids can serve that function as well. All right, so these nucleic acids are ultimately a polymer which gets the official name of polynucleotide, okay? So what I want to do is I want to draw a bracket around our polynucleotide. There we go. Each polynucleotide is made of monomers called nucleotides. All right, so I'm going to circle those for you. Here's a nucleotide. Here's a nucleotide. Here's one. And here's one. We have 4 nucleotides. So what is a nucleotide? It looks like a hexagon, green pentagon, and a circle. Well, yeah, let's blow it up and take a look. Here we go. We have our hexagon called a nitrogenous base. We have a green pentagon called a sugar. And then this little tiny yellow circle is called your phosphate group. So that's what's in every single nucleotide. Nitrogen-containing base, pentose sugar, and phosphate group. Now, let's zero in on those nitrogenous bases and add a little bit more meaning to them. So here is a whole bunch of fancy looking molecules, right? These all have names, and these are your choices for nitrogenous bases in a nucleotide. Cytosine, represented by letter C. Thymine, represented by letter T. One that's in RNA called uracil, which is represented by letter U. Adenine is A. And guanine is G. Now, again, if you've ventured into any biological territory in your past classes and learned about DNA, I hope they mentioned A's, C's, T's, and G's, okay? Because these 4 letters ultimately are what's inside your DNA that code for everything that makes up you and your body. The reason you're you is because you have a specific combination of this chemical, this one, and this one, and of course this one in RNA. So isn't that amazing that the blueprint of life relies on 4 little nitrogenous bases to ultimately build something as complex and complicated as yourself? Awesome. The other nucleic acids have some of the same components as what we see in DNA. Like, for example, ribonucleic acid or RNA. It looks a lot like DNA if you look at it over here, but there are some differences. The sugar in RNA is called ribose, and that's where this part of its name comes from. Where in DNA it's deoxyribose, because the sugar inside of DNA is deoxyribonucleic acid. The sugar is deoxyribose. So the two sugars give them different names. They do have phosphate groups. They do have nitrogenous bases. You can see it's A's, T's, A's, U's, C's, and G's here. There's no T in RNA. And notice the difference though, this is just a single strand. Normally there was the other strand with it, and it kind of did one of these in DNA. But in RNA, it's just the single strand and not the double strand. We also have the uracil present instead of the thymine in DNA. And as I was mentioning before, with the analogy of me as a teacher copying a page out of a textbook 125 times, well, imagine that DNA, a particular sequence, has to be copied 125 times. What would come out is this right here, this strip, 125 times if your body needs it. And perhaps you need 125 working proteins that you didn't have before. That RNA will eventually code for it. The other significant function of nucleic acid is going to be energy related. So here you see this brain kind of flashing up with energy everywhere as it's thinking. And that's due to a result of ultimately what glucose and nutrients turn into in your body, which is ATP. So this is the energy currency of your body. I want you to think of this, and this, by the way, this whole thing here is ATP. And you can see adenine, one of your nitrogenous bases, is part of it. Ribose, the sugar for RNA, is part of it. And then 3 phosphate groups, which also are part of DNA and RNA. But I also want you thinking of this thing as a battery, okay? Like your cell phone battery. We usually talk about having bars on our cell phone, right? And when you have full bars, you got them all. And then if you lose a couple bars, you have a whole lot less. Well, think of these phosphate groups here, these 3 phosphate groups, as your battery bars. So this is triphosphate. 1, 2, 3. There's your triphosphates. And if you use one of your phosphates, then you're down to 2. Your battery's going down. If you use 2 of them, then you're down to 1. And we call it adenosine diphosphate when there's 2, adenosine monophosphate when there's 1, and you're out of energy, okay? So think of your ATP molecule as kind of an energy currency that works just like the battery on your cell phone. Here you go, there's another version of it. Here we get fully charged battery. Here's your 1, 2, and 3 phosphates. And you spend one, and then you got a whole lot less energy. You spend another, you got even less than that. And how do you recharge it, of course? You eat, right? And that will recharge those phosphates, give you more ATP to go do whatever you got to do. Think, breathe, play sports, walk, whatever. Here is what it looks like in chemical structure form. Here's that triphosphate area. If I spend one of these with some, say I'm working out, and now I'm down to diphosphate. And as the number of molecules approach this status, then it's time for a recharge. And you go and you have some lunch and fuel back up. All right, so we've covered now proteins and nucleic acids in this part 2 that go with the carbohydrates and lipids from part 1. Hopefully you learned a lot from this. Take your time, make sure you read the textbook that accompanies this information, and I'll catch you at the next lecture. Bye-bye.